Welcome everyone. Today we're going to be going over the first section of this course. It's actually going to cover 3 chapters here, Chapter 1, Chapter 4, and Chapter 5. So as I go through this PowerPoint here, you're going to see we're going to transition from each chapter as we go, but I'll put timestamps for each of the chapters along the way so that you can skip to certain chapters if you'd like to do that as well. All right, well, let's go ahead and get into Chapter 1, which is just a general overview of how drugs are used and some different things to talk about here in this chapter. So we're going to go over some general principles of drug use. We're also going to talk about some important terms to be aware of here. Talk about some correlates of drug use as well, and then some antecedents of drug use and things that might cause people to be more likely or potentially less likely to use different sorts of drugs. All right, so let's start off by talking about some general principles of drug use here. So number one, I want to talk first about the fact that general use is not necessarily abuse. There's 2 different kind of sides to this, and there is a line that sort of gets drawn when we're talking about abuse. So most users of any given substance really don't use it in ways that can be defined as addiction or as a substance use disorder. So it's also important to remember that every drug has multiple effects here. So although some users might seek only one effect, every drug itself acts at multiple ways and on multiple parts of the body. Also, amount matters, right? So large doses or frequent doses can have larger effects than maybe small or minuscule doses over shorter periods of time, or even single doses at a given time. So that's also important to kind of keep in mind here. I also want you guys to think about that the user's history and expectations are really a powerful influence over the psychoactive drug effects here. So experienced users really react differently than new users. Oftentimes experienced users may report more of the positive effects of a drug and less of the negative effects. And sometimes the expectations really can influence the user's experience. So if they've had positive experiences in the past, they may be more likely to remember those or think that that's what's going to happen in the future. And then, of course, we want to really kind of frame this from the sense that drugs are not necessarily good and they're not necessarily bad, right? So any drug that produces effects might produce some benefit when used in the right way, or has the potential to produce some harm when abused as well. So blaming the substance really can kind of not be the best way to approach this, right? We want to think about all the factors as well as the positives and the negatives, as well as the user that can kind of go into this effect here. All right, so let's talk about some important terms that I want you guys to be aware of and to understand. Number one, obviously, is just drug, right? So this is any substance, whether natural or artificial, other than food, that by its chemical nature alters structure or function in a living organism. Illicit drug specifically is a drug that is unlawful to possess or use. So it's against the law to use an illicit drug. Deviant drug use are drugs or drug use that is not common within a social group and that is disapproved of by the majority. So things that generally aren't accepted by different groups of people. Then we have drug misuse, and this is different here. So this is really the use of prescribed drugs in greater amounts or for purposes other than those prescribed by a physician or say a dentist or something like that. So this is really talking about prescribed drugs used in an incorrect way. Then of course we have substance addiction here, right? So this is the case where people have struggled to control their substance use and have often sometimes had some negative consequences from that use. All right, so those again are some important terms to keep in mind. These are also defined in your textbook. So if you're following along with the textbook, there's some more information there as well. And of course, you can look up these definitions, but these are the important terms to kind of keep in mind as we go throughout this chapter. All right, next let's talk about some correlates of drug use. So what is a correlate? So a correlate itself is basically just a variable that is statistically related to some other variable, in this case maybe drug use. So I put marijuana up here on the list because most research that kind of looks at these drug correlates often, and so far, has really looked at marijuana smoking sort of as an indicator, mostly because there's a lot of data, a lot of use of this drug. And so it's easy to kind of look at how it correlates with other factors as well. And so because a lot of people may have tried this in particular, there's more data and more reference points to kind of look through. Let's also talk about some risk factors and some protective factors of these correlations or correlates of drug use. So what is a risk factor? A risk factor is really things that might lead someone to be more at risk for higher rates of drug use. Protective factors tend to be things that are correlated with lower rates of drug use. So I have some up here on the list here you can see, but so we'll go through each one here. So deviant drug use tends to be, so adolescents who are likely to maybe smoke cigarettes or drink heavily and smoke marijuana are also likely to engage in other deviant behaviors and often tend to not do well in school. So that tends to be a risk factor we can see. Gender also is one that we've seen in the data where males tend to be more likely to use tobacco or smoke marijuana and use cocaine specifically than females are in this case. For race and ethnicity, really some examples here are that white people tend to be more likely to drink alcohol, use tobacco, or use cocaine than African Americans or Hispanics. That's based on some of the data here in the US at least. Level of education, some examples here, young adults with college degrees tend to be more likely to drink alcohol and less likely to use tobacco compared to those with just high school degrees. And then personality can play a role here as well, right? So it might be a smaller role than some of these other factors, but it does kind of come into play here. So again, whether someone decides to try alcohol or marijuana can play a factor here just in whether or not they have other, motives for trying different drugs. Impulsive individuals might be, more likely to use drugs at higher rates. And so what I have here to kind of show you is a table that kind of lists some of the risk factors and then also some of the protective factors here as well. And so these kind of fall in order of importance. So risk factors, again, are those that are going to cause people to be more likely to engage in drug use, and protective factors are going to cause people to be less likely to engage in drug use. So take a look at this table and you can kind of see how some of these play a role here as we go. And then last, of course, I'll just note that genetics does play a small role here. There is still a lot of research and data kind of being disputed on this, I think, but there is some level of genetics that may play a role here, but not as much as some of these other factors. So it's a much smaller piece of the pie here, if you will. All right. So let's talk about some antecedents of drug use. So what are some things that might cause people to be more likely to engage in drugs, or that we can see people tend to do prior to drug use? So antecedents itself is a word, right? Is a variable that occurs before some event, such as the initiation of drug use. So it's not necessarily what causes drug use, but it may be sort of a sign that someone may be likely to use drugs in the future. So let's look at some examples here, right? So some examples of antecedents might be just being generally aggressive, having conduct problems, maybe some poor academic performance, attachment to drug-using peer groups, or even maybe some parent or community norms that tend to support drug use. Now, oftentimes these kind of get misaligned with gateway substances, right? Or a gateway drug. You may have heard that before. Oftentimes it's associated with things like marijuana, but this is really something that's, is only a small factor really when we're looking at some of these effects, right? So when we talk about drugs, remember, drugs are any type of chemical substance that alters the body. So if we look at things like nicotine, caffeine, alcohol, these more common drugs that people are around the world use in fairly high frequency, it's hard to then point figures at other drugs as sort of gateway drugs. So really a gateway substance should maybe perhaps best be thought of as some early indicators of a general pattern of deviant behavior, maybe that results from some variety of psychosocial risk factors, right? So basically the idea that if people are more likely to use some of these drugs, they might be more likely to try other ones in the future. Not that these drugs have caused them to sort of go down the path of more heavy drug use. And then, of course, there are different motives for drug use, right? So different motivations to why people tend to use drugs in the first place, right? So maybe they, again, have some identification with some deviant subculture, some group of people that just likes to, get together and use these different substances. Maybe they have just general rebellious behavior, or there are also reinforcing properties of drugs, right? That sort of keep people coming back, right? They can get things like addiction and things like that. Maybe they have some desire to experience some altered state, right? They're trying to sort of get away from their daily life or sort of escape into this psychological state, or possibly even some of their own experiences that they've had in the past with a drug, maybe some positive experiences that they're trying to relive or search out for. So what I have here is basically some antecedent, some influences on drug use, so you can see here that they kind of break down into some different categories, right? So they have some individual influences, right? Like personality, knowledge, or personal beliefs of drug use. But then there are also community and family aspects as well, right? Does your family often engage in these, behaviors, or your peers, or your local police influence on this situation? And then also we have the larger picture of the societal view of drugs, right? So are there larger, patterns of thoughts when it comes to certain substances? For example, things like coffee or caffeine tend to be pretty well accepted in society, whereas heroin, on the other hand, probably isn't very accepted in society. So all of these levels come into play when someone is being influenced or not influenced to use a drug. All right, so that is chapter 1. So chapter 1, again, very basic, just general overview of drug use, some of the important things to kind of keep in mind and take from it. So we're going to transition now into chapter 4. So let's go ahead and talk about this. So chapter 4 is the nervous system. So here we're really going to talk about, how the nervous system itself works, but also how sort of drugs kind of play a role in this process. So we're going to talk about some different components of the nervous system. We're going to talk about the different types of nervous systems. So that'll make sense as we go through them, things like central or peripheral nervous systems. We're also going to talk about neurotransmitters. So this is kind of the key component here of how drugs interact with the nervous system through these neurotransmitter components. And then we're going to talk kind of through the life cycle of a neurotransmitter, kind of how it goes through the different processes of, just resting in the body to being activated, and then sort of its effect on these different neural components. And then we're going to talk about some examples of drug actions and how drugs actually kind of fit into this puzzle here. All right, so let's talk about some different components of the neural system first. So of course we have to start off by talking about neurons, right? So neurons make up our nervous system, right? And these are comprised of a few different components. So we have a picture of a neuron here up on the screen, and we can see that it has some general kind of similarity structures to it to other neurons we may have seen in the past, right? So we have things like the cell body, that large area on the left-hand side with the nucleus in the center of it, right? From there, we see things like the dendrites coming out. Those are those shorter little orange like roots coming out of that cell body. Those are actually what's going to take in signals from other neurons and send it to that nucleus. Then we have the axon, that long sort of skinny part with the tubes on it that extends out to the right. That's the axon that's going to send the signal from this neuron out towards other neurons. And in the end of that, all the way on the right-hand side, we have what are called those axon terminals. So those are the little pieces that actually touch and connect to the other neurons' dendrites, and that's how the signal gets passed from one neuron to the next. So aside from neurons, we also have other types of cells in our nervous system called glia. So the glial cells are actually cells that are primarily there for many other reasons other than just sending signals back and forth. So they provide different things like they provide the firmness and the general structure to the brain. They help nutrients get into the system, eliminate waste. They also form the myelin. So you can see on that image of the neuron, you see the myelin sheath, those sort of like tubes that wrap around the axon. Those are helped to be formed by different glial cells, and those actually help that signal travel faster down that axon. Glial cells are also helpful in creating the blood-brain barrier. So this is an important component here in keeping out toxins from our brain, right? So it actually helps to filter out and only allow in certain chemicals that are able to pass through and get into the area of our brain that affect these neural cells. And of course, again, the glial is also useful in communicating with other glial cells, right? So they kind of work together to perform these other actions aside from sending these signals like neurons do. All right, so let's talk now about neurotransmission. So how sort of that information gets passed from one neuron to the next. So we already kind of went through how that signal travels from the cell body down the axon to the axon terminals, which connect to another cell's dendrites that then receive that information. But when we talk about this, it's important to keep in mind too that as that signal goes through the neuron, inside of the neuron itself, it's an electrical signal. So there's this electrical component. You can kind of see on the right-hand side, this graph showing up here of how this voltage in the neuron actually changes to get to a point where it can actually fire, right? So we see here as we go through this chain of events that happens in order for the neuron itself to actually fire and send some signal. So we start off sort of at this resting potential, this sort of like neutral state where the neuron is not firing, right? So this happens because there's actually sort of an uneven distribution of ions in that neuron. We don't need to get too much into those details right now, but just know that sort of this resting state is called hyperpolarization. Then what happens is these ion channels actually open up, and we'll see some demonstrations later on of how this happens, where those ion channels will open up and actually allow those ions to move in and change the voltage within the cell. This is called depolarization. And you can see on the graph, it's as that line is sort of starting to go up towards the higher peak. And when that reaches a certain point called the all or none threshold, so either it reaches this point or it doesn't, it has to meet that point. It's either all or none. Then what happens is the neuron reaches what's called its action potential, and it's able to fire. So you can see as it hits that peak in the top, it's able to fire and send off a signal. And then once it does that, it has to repolarize and come down and sort of reset. And in this process, we get what's known as a refractory period, where it's actually so far down that it needs time to recover before it can fire again. And so we see sort of, again, this chain of events that repeats over and over every time a neuron is going to fire. And this is kind of the process that it goes to. And you can also see in this image some of those failed initiations. That's where maybe it starts, but it doesn't meet that all or none point, and so it doesn't actually fire. So the neuron doesn't have any level of strength to its firing. Either it fires at full force or it doesn't fire at all. Those are the only two options it has. All right, let's talk now about the central nervous system and the overall nervous system and the types that we have here, right? So we have the central nervous system and the parietal or the peripheral nervous system, central versus peripheral. And you can sort of see if we start in the top of this tree, we work down and it first splits into either the peripheral or the central nervous system, right? So some of the functions of the central nervous system right off the bat are, this is things like the brain and the spinal cord. So there's a lot of functions here, right? But generally it sort of is designed to integrate information, it's learning, memory, coordination, things like this. that we think of a lot of the time that our brain is sort of responsible for. It's those big picture components. And then when we get into the peripheral nervous system, this breaks down into two general categories as well. So there are a few others, but we're only going to talk about these ones here because these are sort of the main ones that I like to focus on. So first we have the autonomic nervous system, or abbreviated as the ANS, right? And this itself actually breaks up into two different components. So this has both the sympathetic and the parasympathetic nervous system here. So typically the sympathetic branch is responsible for sort of that heightened sense of awareness, or of sort of that heightened sense of activity, right? So we get things like the fight or flight response, increases in heart rate. These are things that tend to happen during that sympathetic nervous system response. On the other hand, our parasympathetic response is designed to sort of calm down our nervous system or reduce different things. It helps to reduce or decrease our heart rate and sort of come back into a more calm state. So think of that as sort of the two different sides there. So sympathetic is sort of that increase in activation or awareness, parasympathetic as the calming or reduction in that. And then, of course, we have the somatic nervous system, which is a separate piece, again, part of the peripheral, but not part of the ANS. And this somatic nervous system is sort of the cells on the front lines, right? So they interact with the external environment. They help control a lot of our voluntary actions, things where we are actually in control, moving our limbs, things like that. They carry out motor or movement function of those peripheral nerves, right? Like, so the extremities of our body, things like that. All right, so next we're going to talk about some neurotransmitters. So these are the actual chemical pieces of the process in converting or sending signals from neuron to neuron. So I said before, actually, that when we send a signal in a neuron, it's an electrical signal. And that is true inside the neuron. But when one neuron sends a signal to another neuron, it actually can only communicate through chemical responses. So this chemical process, which connects the two neurons, uses what are called neurotransmitters to accomplish this goal. So you can kind of see in this image, actually, right off the bat, this is an example of the dendrites and the axon terminal of two different neurons. And you can see they don't actually touch each other, but instead they communicate through passing back and forth these neurotransmitters to send this chemical signal to each other. So one of the first ones we can talk about here is dopamine. So dopamine you may have heard of before, right? So this is oftentimes part of the reward center of our brain, right? Or so often results in a sense of reward or positive outcome when we get sort of dopamine response. We also have acetylcholine. So acetylcholine is often things that can help with like memory or blocking some different memory impairment. So we often see sometimes those with Alzheimer's disease have issues with acetylcholine here in this case. Norepinephrine is another neurotransmitter here we can talk about. So this tends to influence the level of our arousal and attentiveness to things. It can also play a role in how hungry we are, right? Or how much we want to intake different foods. Serotonin is another one that is oftentimes associated with reward or things like that. So oftentimes it has a role in things like impulsivity and aggression, sometimes depression, also food intake and weight control, and also things like alcohol use and just general sort of impulsiveness. Serotonin also, I'll just make a note here, can sometimes play a role in the actions of some psychedelic drugs as well. We'll talk more about that when we get to those specific drugs later on. GABA is a really interesting one. So GABA is actually an abbreviation, right, for gamma aminobutyric acid. So this is interesting in the sense that it actually works to kind of calm down our nervous system, right? So It's sort of one of the ways that we reduce activation when there's overactivation in our nervous system. So it's an inhibitory neurotransmitter. It reduces activation and inhibits that activation. GABA is also often found in alcohol, which is one of the reasons we see alcohol reduce our ability to perform functions or cause impairment. Glutamate is found in most of the regions of the brain, actually. It's an excitatory neurotransmitter, so sort of the opposite of GABA here. And so again, can sort of be helpful in exciting different neural pathways when they need to be excited. And then again, GABA reduces those neural pathways when they need to be reduced. We also have endocannabinoids. So this also is found fairly distributed throughout the brain. A lot of recent research has sort of looked into this. And this is also tied to things like marijuana and can play a role in this neurotransmitter process. We also have endorphins, again, throughout the brain. These are naturally occurring, similar. They have like a morphine-like effect. So they actually play a role in pain relief, as well as feelings of euphoria and other general functions. All right, next let's talk about the life cycle of a neurotransmitter. So I gave you some examples of what each of those were, but now we're going to talk through kind of what the process looks like and what their cycle or their life cycle actually is. So step one is just that they are kind of existing, floating around in our bloodstream, right? In the brain. They're just generally circulating in the blood, available to be used. Step two is they actually get what's known as uptake, right? So they get taken up by cells to be then used as part of this communication process between neurons. Then we have what's called synthesis, right? So these are precursors and synthesized into neurotransmitters through the action of enzymes. So they actually get transferred into neurotransmitters through this synthesis process. And again, enzymes help to, undergo that process and actually change the molecular structure to then make them usable as neurotransmitters. We then have storage. So now that they're available to be used, they get stored in sort of these what are known as synaptic vesicles. You can see in that image on the right, there are those bubbles sort of in the top part. And they store them there in the axon terminal, ready to be released to send that signal to the next neuron. Then we have the release, right? So when they're ready to be used, when that nerve is ready to fire, it releases those neurotransmitters out into what's called the synapse. That's that empty space between the axon terminal and the dendrite that's receiving it on the other end. So there's this gap, this empty space, and they need to travel through that. And again, that's called the synapse. So they're released into the synapse and they float around and are then in step 6 used in binding, or they are bound to those receptor sites on the receiving dendrite of the next cell. So you can see sort of those little, almost like trees, the blue and green, they're shaped in a way that they accept those neurotransmitters from the synapse and use those to bind to those receptor sites. And that tells that receiving neuron, hey, okay, I'm receiving this signal. It's my turn to now determine if I'm going to fire or not. And then, of course, we have metabolism, and this just helps to break down those neurotransmitters back into sort of their, resting state, to be put back out into sort of our bloodstream to be started, able to start the cycle over again. So that metabolism just kind of breaks them down back into their resting state. All right, let's talk about some examples now of drug actions and how this kind of works in practice, right? So here we have sort of what are known as both agonists and antagonists. So these are two different types of drug actions that sort of work as drugs are administered into this process, this life cycle of the neurotransmitter. So agonist or an agonist drug tends to mimic some neurotransmitter, right? So the actual chemical of the drug may mimic some neurotransmitter in the brain and sort of acts to increase the number of those neurotransmitter functions that are present. So it activates receptors just like the neurotransmitter would. And so some examples of this here are like if there are not enough neurotransmitters, but you ingest a drug that mimics those neurotransmitters, you could see an increased effect of what that naturally occurring drug may do. Then we have antagonists here, which act in a different way. So rather than mimicking the neurotransmitter to bind to those receptor sites and activate that response, they instead block the receptor sites. So they may sit inside those receptor sites, but they don't activate them. They just sit there to block out the ability for them to absorb other neurotransmitters that they may otherwise want to absorb. So again, antagonist acts to reduce those signals passing back and forth between the neurons. Agonist increases those signals, antagonist reduces those signals. So an example here too are things like antidepressants, right? So these may block receptors. So these may block reuptake, actually, sorry, not receptors, block reuptake of things like dopamine and serotonin and norepinephrine. So if they're not able to be, reuptook into the into the synaptic, into the axon terminal, then they can't be reused, and they stay in that synapse, and they continue to bind over and over again and continue to fire more and more. So it increases the ability of this binding of these two different factors. All right. We will talk more about this as we go as well. When we talk about each drug, we're going to talk about exactly how it works as either an agonist or an antagonist and demonstrate sort of the process of this life cycle of the neurotransmitter with the drug's influence on it. So you'll see how specific ones work as we go. All right, on to chapter 5 here. So this is the last chapter of this section. We're going to go through chapter 5 here and wrap up this section. All right, so chapter 5 is the actions of drugs. So here we're going to talk about some different categories of drugs, just in general. We're going to talk about some drug effectiveness, right? So how effective these drugs can be in different situations. Some different routes of administration, how we can actually get those drugs into our body. And then some different mechanisms of drug actions as well. We'll also end here with some different factors of tolerance and withdrawal on drug use. All right, so let's start off by talking about some different categories of drugs. So we're going to break these down and talk about them as we go throughout the semester as well. But the first category here are stimulants. So stimulants drugs tend to produce some general wakefulness, a sense of energy, well-being. General examples of stimulants are things like cocaine, amphetamine, or caffeine, right? We can see that here in our example. We also have depressants, right? So depressants tend to slow the nervous system activity, reduce sort of that activity. Some examples of this are things like alcohol, benzodiazepines, sedatives, sleeping pills, things like that. Then we have opioids or narcotics. So these tend to produce some sort of relaxed or dream-like state here. Some examples of this are things like fentanyl, codeine, heroin, oxycodone. These are all examples of opioids and narcotics, some of the most probably addictive and dangerous category of drugs right here. Then we have psychedelics. So psychedelics here, of course, things like LSD, DMT, psilocybin. Psychiatric medications, right? These are often prescribed specifically by psychiatrists and other physicians to treat different symptoms of mental disorders, right? So we have things like lithium or other examples that fall into this sort of unique category. And then, of course, here we have some examples that tend to fall into their own category because they have similarities or overlaps with other of these major categories. So some examples are things like nicotine or cannabis or cannabinoids, which tend to fall into their own category because they mimic a lot of the factors of these other categories. So yeah, just kind of showing that here again. They have some effects of maybe psychedelic or depressant or stimulant, all kind of mixed in their own unique way, so they don't fall nice and neat into one of the other categories. All right, so let's talk about the drug effectiveness, or how drugs can be effective under different circumstances. So we have what are known as non-specific effects, right? So these tend to be effects that are unique to the user's background or particular perception of the environment, right? We sometimes call these placebo effects, right? Because they can be produced, by not actually having the chemical, but they still may believe that they are experiencing or ingested that drug. Oftentimes you'll see these with different studies that have been done looking at like non-alcoholic beer on participants' perception of being intoxicated, or just some other general placebo effects that occur under different drug use trials. These are, though, important oftentimes when we look at treating pain and depression. So I think there's research on some of these effects on different chemicals, especially things like CBD more recently, and how this can still be beneficial, even if not directly related to what are known as specific effects, which tend to basically depend on the specific presence of a chemical at certain concentrations in the target location, right? So these are more what you'd think of when you think of those very specific chemical effects that are happening from those drugs themselves. When talking about this, I think this is a great place to also discuss double-blind procedures. So with placebo and testing these non-specific versus specific effects, we look at this often when in clinical drug trials, and it's hard sometimes to break these two things apart if you don't do what are known as double-blind procedures. So these are procedures where not only does the participant not know if they're getting the drug or the placebo, but even the researcher doesn't know which participants are in which group. So it's documented, written down somewhere, right, so that they can find out after the fact, but there's no bias being presented from either the participant or the researcher in these cases. So it's kind of a nice way to ensure, again, no bias gets presented here. So what we have here, and in this image that's up over here on the right, is what's known as the dose response curve, right? So this is a graph that depicts basically the relationship between the drug dose and the resulting drug effect at those different dosages, right? And so what we get here are what's known as thresholds. So thresholds are basically the lowest dose at which some effect can be observed. So we can see based on the different things here, like we have slowed reaction time, ataxia, and then finally unconsciousness, and this is for alcohol, of course, we can see that those effects tend to appear on this curve, right? This dose response curve based on how effective or how much of the dose they've gotten, and then how much of that response we tend to see. And the threshold is basically at what point on that curve does this effect become observed at the lowest dose. All right, and so what we see here too is this idea of how we can estimate the effectiveness, we'll say, of a drug. So at what point do we get these effects that we're looking for while also keeping in mind the safety factor as well? So there's what's known as safety margins of every drug, and these take into account both the effective dose, right? So that is the amount needed to be taken in order to see the effect you're looking for, as well as the lethal dose. So that's how much of the drug at which point becomes lethal to take. And so what they've created is what's called a therapeutic index. And this is, you take the lethal dose and you divide it by the effective dose and you get some value. And that value should be over 1, right? If it's not over 1, then it doesn't do anything except kill you. But if it's over 1, and by how much over 1 it is going to tell you how dangerous that drug is. So how much do you have to take? If it's a small difference between how much you have to take to see an effect and how much you have to take that will kill you, that small difference means it's a pretty dangerous drug. We want a large gap there. We want to be able to have plenty of room to see that effect of the drug before we start reaching the limits where it becomes lethal. And so we can see here actually this chart, which isn't exactly what we're looking at, but I thought it fit well here to kind of depict some of the different values that we have on the active versus lethal dose. So you can see as we get farther over to the right side of the graph, we see drugs with these higher therapeutic indexes, meaning they tend to be more dangerous because they're more likely to overdose on. You can get closer to that lethal dose as you look for that effective dose. So you can see heroin being up in the top, one of the higher ones, and then looking down at some of the lower ones like LSD, marijuana, tend to have a very large margin between the lethal dose and the effective dose. We can also talk here about potency, right? So potency is just generally the amount of a drug required to produce some particular effect, right? So different drugs have different potency. Some may be more potent than others. And then of course we have side effects here as well. So these are the unintended effects that accompany some therapeutic or desired effects that we have of the drug as well. All right, we also have this time course component when talking about drug use here. And the time course, or how long it takes for the drug to get into your system, to be absorbed, to actually become effective, varies from drug to drug, right? So it can also vary within one specific drug. So it may vary based on how the drug is administered, right? So if you do one method of administration, you might get that drug into your system much faster than a different method of administering the same drug. How rapid the drug is absorbed, this varies from drug to drug. So lipid solubility is how much that drug dissolves in oils and fats, and that can affect how long it takes that drug to be absorbed into our bloodstream. And then also how quickly the drug is eliminated from our body. So the longer it takes for it to be eliminated from the body, the more it may stay active in our body as well. And we can see sort of, this model of how we might approach this, where we see an increase in the amount, but we don't really see the measured effect of the drug until a certain point, right? So we might be building up, and then not at a certain point until around spot B there do we actually see an increase in the effect of the drug. And then we see that same sort of decrease effect as we go down as well. And of course, we can prolong the effectiveness of drugs by administering additional doses during this time course, right? If we continue to administer throughout the process, we end up prolonging that effective period. All right, so let's talk about some different routes of administration, because we just kind of mentioned that these can affect how we get those drugs into our system, but also how fast it may affect that effective rate or noticing the effective side of the drug. So of course, first we have oral administration, right? So oral is just ingesting something through your mouth. And some of the things with this is whatever the drug that's being administered orally, it has to be able to withstand the digestive process, right? So it can't be sort of broken down by the acids in your stomach and things like that. They also have to be able to sort of pass through more of your digestive system before they get absorbed into the bloodstream. Typically, only lipid soluble or very small water soluble molecules tend to be able to work in this way. And of course, these tend to also pass through the liver as they're kind of working their way out. So that's another important factor when talking about sort of the consequences of different methods of administration. We also have intranasal, right? So this is basically ingesting something through your nose, right? So it is absorbed through what's known as the mucous membrane in your nose, and it goes into the bloodstream fairly rapidly, and it often bypasses the liver. So this doesn't affect the liver, but instead just goes straight into your bloodstream. Typically, the drawbacks associated with this tend to be things like nasal necrosis or the death of general cells in that region. So it can have some negative side effects, even though you might be thinking it's better because it's bypassing the liver and things like that. Then we have intravenous, right, or IV injections. So this is where the drug is basically injected directly into the bloodstream, right, through a needle, right into a vein, into that bloodstream, right? And so this tends to result in a pretty rapid onset of its effects, one of the faster methods of administration, and tends to be a less rough way to administer it if the drug is maybe more irritating to different senses of your ingestion, right? So it might be a way to ingest drugs that are harder on the system to take. Also, high concentrations can often be delivered this way as well. Of course, this does have some risks, things like veins becoming damaged over time, or of course, bloodborne diseases are very easily spread through this method. Then we have subcutaneous or intramuscular injection. This is basically where the drug is ingested right under your skin, also with a needle, but it doesn't go into a vein, right? It goes into just sort of the muscle or the general skin tissue in that region. Again, you can put larger volumes oftentimes in this way because they get absorbed a little bit slower, but there are some negatives to this too, especially with some more dangerous drugs where you can deteriorate a lot of that skin tissue as well. But plenty of less dangerous drugs are administered this way as well. Think things like insulin or things like that. Then of course we have inhalation, right? So that is just breathing in through the lungs the drug itself. So oftentimes these drugs need to be in some kind of vapor or combustion smoke form, right? This tends to be one of the faster ways of administering that drug as well. So it's a pretty quick way to get the drug into your bloodstream. But of course, risk factors are mostly on the lungs, right? So tends to involve a lot of issues with lung cancer and things like that. And then lastly, we have here is sublingual. So this is when you place the drug underneath the tongue, and this is where it gets dissolved directly into the bloodstream, again, through sort of this membrane. Fairly quickly, fairly fast way of administering the drug as well. So what you see here, again, is a table that kind of outlines the route and some advantages and disadvantages of that different route of administration. So you can see, again, we have oral, intranasal, and intravenous injections here, some of the pros and cons of those different methods. And then on the next page, again, feel free to pause and look at this. On the next page, we have subcutaneous, intramuscular, smoked or inhaled, and then sublingual here as well. Again, advantages and disadvantages of these different methods. All right, wrapping up here towards the end, we've got a few more things to talk about. We're going to talk here about the mechanisms of drug actions, right? So how the drugs sort of have an effect on the neurons. So drugs in general often act through this sort of semi-permeable membrane of neurons, right? So while there's this electrical component, remember drugs are mostly interacting with sort of that chemical side of the neural communication. And they alter the availability of those neurotransmitters. Right? We talked about this in both the synapse as well as those different sites. So we can look at things like synthesis, metabolism, as well as the release from the vesicles and the reuptake into the neuron itself. So these are all different ways that drug can affect or act on this communication from neuron to neuron. Also here, I think it's important to talk about different drug interactions, right? So obviously each drug does not just act on its own. There can be different interactions when taking multiple drugs at once. So this is important to keep in mind as well, that combining different drugs can have positive or negative consequences depending on the outcome desired. So for example here, right, methamphetamine lessens alcohol-related performance disruptions. Alcohol lessens methamphetamine-related sleep disruptions, but they produce greater increases in heart rate and euphoria. So sometimes taken together, they might appear to have a more positive, experience, but they might lead to more increased health concerns and risk factors. All right, and we're going to end here talking briefly about tolerance and withdrawal, of course. So when we talk about this, we have to talk about, in terms of tolerance, how we build up tolerances to different drugs. And that happens in kind of two different ways. So we can talk about sort of the drug disposition or the pharmacokinetic tolerance. This is really where we talk about that physiological tolerance to those different neurons or neurotransmitters in the brain. So this is typically where we see the actual physical side of the tolerance build, where we need more and more of the drug physically to elicit the same response as we did earlier on when taking it. We also have what's known as behavioral tolerance, right? So we tend to see the same biochemical effects, but a reduced effect on behavior. And so that reduced behavioral effect is really a result of the users learning to compensate, right? So they get more used to that, feeling used to those sensations, and they compensate more on how they feel when doing that. And so in that process, they take more of the drug, even though it's having a fairly similar effect physiologically. And this is often how things like overdoses can occur. And then, of course, lastly here, we have the pharmacodynamic tolerance, right, which is basically where we have that overall reduction in effectiveness of the drug. And this is often also what we see that causes those withdrawal symptoms, where our body is used to and needs those effects because that is our new normal. And without that drug acting on our nervous system, we are in the reduced state or the opposite state of having the drug itself. And then this is sort of, again, where we get those withdrawal symptoms and where we see those negative side effects of trying to wean off and get our body back to a normal state where it can function properly without the use of those drugs. All right, so that is section 1, chapters 1, 4, and 5. Again, I'll put timestamps for each of them if you've been following along this whole way. I thank you. Otherwise, that is the end of this section.